Thank you. It's lovely to be here in Paris to talk about my work. So I'm an artist. I'm just going to talk about it from the point of view of the art that I've been making. And they asked me to say what I do, how I do it, and why I do it. So I'm going to try and do that. I'm kind of obsessed with bacteria. That's probably a good starting point. I'm obsessed with these strange organisms, and I've been trying to develop over quite a few years something that I call a bacteriocentric view of the world, because I think as an artist, in order to work with these kind of living organisms, you kind of need to understand the world from their point of view. There's a lot of discussion about, you know, how can we change bacteria to make new materials, or how can we do various things with them? Can we get them to do this? Can we get them to do that? But if you don't understand what they want, what their desires are, and what their needs are, you can't get them to do anything. They won't grow in certain places, they'll die quickly. So we need to kind of have this bacteriocentric view of the world. And that was an idea proposed by Betsy Dexter Dyer in her book, A Field Guide to Bacteria, which I really recommend. It's a book that tells you how to spot bacteria in your everyday environment, on statues and in the ground and things like that. And you'd be surprised how many bacteria are surrounding you. I was going to say, there are more bacteria on the end of your finger than there are people in the world. There are 10 times more bacterial cells in the human body, and we carry around at least 2 kilograms of our body weight is bacteria. 2 kilograms. And we need them, otherwise we would die. They digest our food, and they help us in many ways. But we don't live in a balance with them. We live in a precarious battle with them. So it's wrong to think of this sort of beautiful idea of balance, I think, with bacteria. And I was very interested in the way that there's two ways of looking at bacteria, that you get the impression through the media that it's basically synonymous with dirt. We could buy hand washes and sprays and things to clean our houses, and then we could get rid of bacteria because bacteria are bad. Less than 1% of bacteria are actually harmful, pathogenic to humans, and typically 300 or so strains of bacteria will come through a normal hospital lab that they would observe in the population. But the rest of them, sort of 99% of bacteria, just aren't even studied. We don't look at them. We don't really have any interest in. They're considered to be kind of no commercial or medical interest, is a term that's used. But there is an interest out there, and there's lots that we need to understand about them, because they control the atmosphere of the planet. They produce, through rice paddy fields, 10% of greenhouse gases are created, roughly 10% of greenhouse gases are created through the bacterial reactions going on in when they grow rice. So I started to look at things like that and those sorts of stories, and I started to look at the bacteria in my own home and started to map it. And these are some early examples. And that piece is an ongoing collaborative crochet work that when people come to the exhibition, I exhibit it over and over again. And when people come, there are balls of wool there, and they sit down and crochet with me, and we talk about their perceptions of bacteria, and they help me crochet the bacteria representations of the bacteria on my bed. And that was an early work, but I've been building up on it because I think it kind of models quite nicely how bacteria grow. If I feed it with wool, if I feed it with the correct substrate of the gallery space, and I supply it with enough food of people to kind of help produce it, it keeps growing. I've worked a lot with bacterial communication. So bacteria actually do a kind of thing where they talk to each other. They're all talking to each other all the time. So you've got these 2 kilos of bacteria in your gut. They're all talking to each other. And they can also talk to human and plant cells and animal cells. So some bacteria, for instance cholera, can actually change the shape of the lining of your gut and make your body more hospitable to them. And they can send other signals, and we don't fully understand what's going on. It was only discovered about 12 years ago that they're doing this. But they can do other things. They can actually control or have an effect on whether you feel happy or sad, and all sorts of things like that. That's to do with your gut bacteria. Anyway, I was very interested in bacterial communication, and I found I was working with a strain of bacteria called Chromobacterium violaceum, which turns purple when it's sending and receiving communication signals called quorum sensing signals. And then I had a genetically modified strain called CV026, which was in effect mute. It couldn't send communication signals, it could only receive them. So the normal unmodified strain would be white if it wasn't receiving a signal, but because it's always sending and receiving signals, it's always purple. But the other strain is white. But if you put next to the purple strain, you put the white strain, you can watch before your very eyes, like just moving across it, you can watch the white bacteria turn purple. You can actually observe bacterial communication happening. So I made this this dress. I work a lot with historical textiles because I'm very interested, and I can only say this very briefly, but I'm very interested in sort of the role of women in science and during the Enlightenment, when things like embroidery were considered to be the kind of pinnacle of achievement for women, and women were considered to be part of nature rather than something that could study nature. So the dress was made by inoculating it with bacteria, pigmented bacteria, in the process of communicating. And as the communication signal was travelling across the textile, I had to remove the textile from the agar, and I sterilised it using a technique basically based on pasteurisation. So I heated it to sterilise it without destroying it. The reason for this is it's GMO bacteria, genetically modified bacteria, and you cannot show that outside a lab. You can't show it in a gallery alive. You're not allowed to take it out. You can only show it in a license for GMO Category 2 biosafety lab. So that was what happened with that. But I wanted to bring back that kind of feeling of life to it. So I used a time-lapse film of the bacterial communication taking place, which was video mapped back onto the dress, and I worked with Alex May, who's here today, to do that. So the bacteria were still there, but they were traces of the dead bacteria. I've made a quilt with, I'm an artist in residence on the Modernising Medical Microbiology project, which is Oxford University in Public Health, England, and working with them to kind of really investigate clinical microbiology, and they're looking at how to implement what's called whole genome sequencing of bacteria in the health service settings. So instead of taking a long time to do traditional microbiology, they can extract the genome of the bacteria that somebody's suffering from a condition, and you can get the full diagnosis and basically the prescription that they need to be treated with by looking at the genome, because you can see exactly what drugs the bacteria is resistant to or would be susceptible to from the genome. So they're looking at that, and I was working with them on ways to kind of widen audiences as well for their work, and they're really interested in bringing the public into the story about their work, and having a different kind of interface rather than just disseminating their research through scientific papers. They want to get it into different settings, so ordinary people who changes in healthcare are very important to, can actually have some input into it. So I made a quilt with MRSA bacteria.
MRSA, c'est le staphylocoque doré qui est connu pour sa résistance à l'antibiotique.
And there's patented chromogenic agar, so colour-changing agar, which changes colour in the presence of MRSA bacteria, and it turns blue. So each of the patterns on the quilt were made using different tools and techniques in the treatment and diagnosis of the disease. So you can see on there the dots, the sort of polka dot shapes. They're vancomycin susceptibility discs. And that's a section of the quilt after it was finished. That's the stripe, there's cafoxetin antibiotic, and up at the top there, the yellow embroidery on there is stained with turmeric, the spice turmeric, which has a limited effect on MRSA. So I was combining natural dyes and things with clinical antibiotics as well. And I've done quite a few things with that. And then this dress, which was shown in Capelice Gallery last year, is made with VRSA, which is, I don't know, have you got a word for that? Yeah?
VRSA, just a minute. I don't know.
It's vancomycin.
C'est un staphylocoque doré résistant à la levée.
Resistant to vancomycin.
Vancomycin, donc qui est un antibiotique.
So it's resistant to the very last ditch antibiotic that they currently have available for it. So if this came into your hospital lab, you would be very scared if it came in from a patient. I had access to it through the Public Health England lab, where it's a research thing, but we have to be extra careful when we're working with it because it is highly drug resistant bacteria. And then the rest of the dress was also decorated with the bacteria that is actually cultured from my nose, because I'm very proud to say that I have 2 different strains of Staphylococcus aureus living in my nose, one off each side. And at least 30% of the population carry this bug as a kind of normal skin flora. And it could be that we all carry it from time to time. It sort of appears and emerges. And it's not really harmful to us unless we're sick. MRSA and VRSA are particularly attuned to the ecological niche of the hospital. So they've actually evolved that they're not very harmful to healthy people, but they're very harmful to sick people who are in hospital. They've developed a way of producing biofilms that will coat, like when they've got the lines in your arm if you need a drip in the hospital. They're very good at coating things like that, so they can get into the body that way. But a normal immune system would fight them off quite more easily than it would an ordinary non-drug resistant Staph aureus. And I do participatory workshops like this, and over the last 2 days I've been doing workshops here with people, teaching them how to develop a bacteriocentric view and doing relatively simple microbiology techniques, but with the aim that they could start to do it themselves from what I've taught them, because there's quite a barrier to entry with microbiology. It's if you're an artist interested in it, how do you get into that world? And so I've given them quite basic techniques and ways of looking forward and understanding what they're doing, so they could take it forward in future on their own. And I've been working most recently with Mycobacterium tuberculosis, which is the strangest organism really in the world, I think, which causes TB, which infects 1/3 of the world's population currently, which is a huge number. It will make 10% of those people it always makes someone cough, just the mention of TB. Everyone starts coughing. It's always funny. And it infects 10% of those will get actually sick from it, so most of them are just carriers, and then 5% of those people would die without treatment. But obviously we have treatments for it, but it's becoming increasingly drug resistant, and it takes sometimes a cocktail of between 3 and 6 antibiotics to cure it. But I was really interested in the history of it, because we've struggled with this disease over so long, and they used to think, I found one document from 1902, which basically it was the advice of the National Society for the Prevention of Consumption in the UK, that just stated where there's dust, there's danger, and that dust was the very main cause of TB at the time, they believed. And I found other texts, even from the 1930s, where they don't believe that Mycobacterium tuberculosis, the bacteria that does cause it, caused it. So I made this piece, which it's a combination of needle felt, which is kind of wool that's not been spun, which you can stab with a needle to sculpt it into various forms, but mixed with dust. But the dust had been impregnated with the extracted DNA of tuberculosis, which I had to do in a category 3 lab, and it's a it's a relatively new protocol available in the last year to prove that you can kill TB, properly kill it, in order for me to be able to take it out of the lab. They didn't do it for that reason. It wasn't just for me. That would have been a very expensive art project to study that. Stop laughing, Annie. But it's a byproduct of the research that they're doing, because they want to be able to do whole genome sequencing of tuberculosis, and that to do that properly you need to be able to take it out of a category 3 lab, which is very restrictive. You have to have a, you know, its own airflow, its own air supply, sealed floors, and lots of very complicated things to do it. But they wanted to be able to do it in a normal environment with, you know, with machines in an office or something like that. So using this protocol, they proved over, they tried to culture it over 50 times. Public Health England did it, and Oxford University did it, and they proved it's a valid kill protocol. So I've now exhibited for the first time the killed DNA of, the DNA of killed tuberculosis as part of the installation. And the forms on the lungs there that you can see represent different treatments and different effects on the lungs. So there are granulomas, so TB tends to kind of get into parts of the lung up here, normally on this side. And they would also collapse lungs actively to try to cure people of TB. They thought that this was an effective method. This was a fantastic public health thing at the time. This is a blue Henry, which is a sputum cup for people to spit into if they had TB. But on the top of it is a transmission map that was published in The Lancet last year by the Modernising Medical Microbiology Project that demonstrates the existence of what's called a super spreader, so that one person's responsible for spreading the disease to a lot of people. But the difference between this and the way they discovered the super spreader, kind of Typhoid Mary, is that they didn't do any interviews with people, they didn't do any traditional epidemiology. They got the bacteria from the collection in the lab, they sequenced it, and from that they could tell who gave it to who. And they've created it. It's a whole new way of kind of looking at bacterial transmission. And it's a revolution in kind of healthcare. It's very complicated in terms of the bioinformatics approach, and my next project will be to look more deeply at the bioinformatics and the algorithms behind it. And I've been working with them for 3 years now, and I think I'm just about ready to start tackling that. The microbiologists on the project say, well, we don't even really understand what they're doing. So it'd be great if you can kind of try and struggle with this and kind of make a bridge between that as well. And I think it's really important for the public to be able to understand kind of what's going on too. So it's about kind of communicating it in a way, but also bringing in those really kind of emotional sides of it. So with, and I think it's really interesting for our artists to be embedded long term in research projects. And one project that I'm doing with Lucas Evers that's speaking later is called ICT and Art Connect, and our aim with that is in the future, in sort of European projects and things, to encourage the European Union to put artists long term into research projects. So not just a 1 month thing with a small amount of funding and then you have to do an exhibition or something like that, but to have an artist in the role for 3 years, 5 years, slowly trying to understand the research, responding to the team and becoming a full member of the team would be our aim with it, which is kind of what I've been doing with Modernising Medical Microbiology. I'm also really obsessed with natural dyes, and there's a huge history, which I can just touch on now, of the relationship of natural dyes and antibiotics. So this object here, which represents the idea that rest being the main cure for TB until streptomycin became available. The hospital screen there's dyed with madder root, which is the oldest dye known to man. Turmeric's also antimicrobial. Woad is antimicrobial. Walnut husks, these are all traditional natural dyes. Safflower, again, has antimicrobial properties. And there are herbs that can interfere with bacterial quorum sensing. Oregano, for instance, the herb, can stop bacteria from being able to transmit communication signals. So there are lots of interesting things we can do in very do-it-yourself kind of approaches as well. We can do just in our sort of home experiments. And this dress that I made, it's actually dyed with walnut, and the embroideries on it are with madder root and safflower. It's a maternity dress from the Romantic era, because there's this huge correlation with tuberculosis and art. there's an idea called the spes physica of, in late-stage pulmonary disease, Galen, the sort of ancient doctor, reports that patients, however sick they were, would not accept that they were dying, and they had a huge kind of voracious need to work. And a lot of artists and writers particularly are known for have completed kind of great works whilst suffering from this. The most notable, I guess, is George Orwell, who finished Animal Farm and died 3 weeks later of tuberculosis. And he's really suffered with tuberculosis, so it's not that well known. And one of the treatments that they performed on George Orwell was pneumothorax, and this is a pneumothorax machine that I've acquired and I've changed, I've carved and engraved. And it was used to collapse the lungs of unfortunate patients in order to give the lungs a rest. And this was one of these very strange treatments that they used to undertake for tuberculosis. There's a lot more about it on my website, so if you're interested, I don't have much time now, so I'll just go through these. I think this is the last one, but just to finish up, by kind of being really interested in this stuff and trying to sort of deeply engage with the scientific side of it, I've managed to get access to some very interesting and strange locations, and I think I'm the only artist that's ever been allowed into Porton Down in the UK, which is the highest security lab in the UK, which was built in the late 40s as the UK biological weapons facility. But a year and a half after it was built, the UK signed the bioweapons treaty. So now they don't make bioweapons there. They just study things that could potentially be used as bioweapons. And I was actually invited, and it was a fantastic opportunity, slightly terrifying, to participate in a workshop that they ran. It was just before the British Olympics, the London Olympics, which was a course for microbiologists to prepare them for the Olympics, but in identification and handling of what they called potential deliberate release category 3 agents. And so I was actually able to handle things like Bacillus anthracis, which causes anthrax, and in this case, Yersinia pestis. That's me holding a petri dish of Yersinia pestis, which causes plague. And they taught me how to identify them, how to handle them in a category 3 lab. Don't worry, I've washed since.
I hope so.
But it's funny because you get to come out of the lab and you still have the same jeans on that you wore in the lab and things, and you're sort of looking and thinking, should I have... We had to drive home, and I was like thinking, Anyway, so it's taken me into some very strange worlds, and the scientist that I work with most strongly is Dr. John Paul at Public Health England. And recently he was asked to give an interview for the Guardian newspaper. And I overheard him saying to the Guardian reporter, it's a big newspaper in the UK, he says, well, of course you have to do public engagement in science now, don't you? And I looked horrified and I said, don't say that to her. And he said, but what I mean is it's the perfect excuse for me, isn't it? So now I can just do it, because nowadays you have to do it. So I've got the perfect excuse. And he also came to me and said, he said, it's so funny, the Modernising Medical Microbiology project, they do all this research on Clostridium difficile and different genome things. And he says, but your art exhibition's got more publicity than any of it. So it's like, in terms of actually getting their message out to people, it's reaching a very different audience. And on Saturday, The Lancet, which is one of the world's leading medical journals, published a 2-page article on the exhibition, the tuberculosis exhibition. So it's kind of made it into the medical world as well as the art world. Maybe sometimes more into the medical world, but you know. So I guess that's kind of it, really. Yeah, that's my websites and things. I'm on Facebook and Twitter, if you want to follow me. I also have an Institute of Unnecessary Research, which is artists who work with, and artists, scientists and philosophers who work kind of outside the bounds of normal research, or what you'd normally consider fundable research in unusual ways. And that's it, really. Thank you.
Thanks a lot.